So now we're going to talk about transport across the cell membrane. So far, we've spoken about the cell, we've defined the cell, we've talked about the general structure, and we've talked about all the cellular organelles. So now, like I said, we're going to talk about transport across cell membrane. So I've divided transport across cell membrane into parts A and B. So part A has to do with transport according to the molecular structure, or transport molecularly, and then part B has to do with transport in accordance to or related to the membrane, the cell membrane. So in part A now we have two divisions: passive transport or diffusion and active transport, and each of these have their separate types. So part A division, actually, since it is based on the molecular structure of the components that are going to be transported, it has to do with selective permeability and the channels of the cell membrane. So as discussed earlier, the cell membrane is actually permeable, selectively permeable to particular molecules. It is freely permeable to oxygen and carbon dioxide, but it is not permeable to molecules like water and glucose. Water, because it is not liposoluble, it is lipophobic, and then glucose because it's a large molecule. So molecules like carbon dioxide and oxygen will be able to pass through this selectively permeable membrane, but then molecules that can't go through the selective permeable membrane go through channels, and then these channels can be open or gated. So open means that they are open they are open at all times, every single moment in the cell they are open. Remember, channels are created by our integral proteins. And then gated means they can be closed, which by like a hinge like a door. So it means some can be open and some can be closed. Okay, so for the first one under our molecular division is passive transport or diffusion. So from our elementary knowledge, that is physics or chemistry. So diffusion is the movement of particles, the movement of molecules from a region of higher concentration to a region of lower concentration, in this case through a selectively permeable membrane or through a permeable membrane. So the passive transport, number one, it requires no net energy, no use of energy. There's no use of energy in passive transport, but the only energy, the little energy that can be used, which is not considered in this case, is thermal energy gotten from the movement of those molecules, but no energy is used in passive transport, number one. Number two, passive transport goes down or along the concentration gradient. The next thing we're going to talk about is going to be facilitated diffusion. So facilitated diffusion is also going to have most of the qualities of simple diffusion, being that one, no energy is going to be used, that it is going to also move down or along the concentration gradient, but in facilitated diffusion there is use of carrier protein. So that's what distinguishes it from simple diffusion. There is use of carrier protein. So what does that mean? That means that it doesn't support, or it cannot pass through the membrane of the cell, which means that it is either that molecule is bigger or is too big to pass through the cell membrane, like these little holes in the cell membrane, or it is not liposoluble. So examples of things or molecules that can be transported via facilitated diffusion is glucose. For example, our glucose transporters, GLUT1, GLUT2, GLUT3, and GLUT4. GLUT1 is majorly in the blood, that's red blood cells, and then via its transport via the placenta or in the placenta, and then GLUT4 is majorly adipocytes or muscle cells. So now osmosis is the movement of water. It is the movement of water from a region of higher concentration of water to a region of lower concentration of water. Osmosis is not the movement of solute, it is the movement of solvent, which is water, from a region of higher concentration to a region of lower concentration. There is still also the same down slanting gradient, so it is still classified under this. So there is no use of energy, it is moving down concentration gradient, and then there is no carrier protein in osmosis. So one very important thing to note in osmosis is that it actually has a very, very important clinical significance. Very, very, very important, very relevant clinical significance. Okay, so now let's come here. Just this. We're just going to look at this right now. Okay, so now one of the cells that are majorly involved or majorly undergo this osmosis transport is the red blood cell in the blood. So as we know, our blood comprises of cellular content and liquid or fluid content. So the fluid content is majorly plasma, and then the cellular content is majorly our red blood cell. So since osmosis is the movement of particles from movement of H2O, fluid, liquid, water, from higher concentration to lower concentration through a membrane. So this is an isotonic red blood. This is a red blood cell in an isotonic solution, in which the solute content within is solute content out. Here there is no transport of H2O in or out of the cell. But in the event that our plasma has a high solute content, high solute content means it has low H2O content. If it has high solute content, that means the H2O ratio is low. That means that our plasma is very, very concentrated. So what is going to happen is that water is going to move from a region of higher concentration. That means the cell, the red blood cell, will have the higher concentration and the plasma will have the lower concentration, since it is high concentration in solute. So water will move from the cell into the plasma, and that's going to cause plasmolysis, and it's going to cause shrinkage. Okay, that's a hypertonic solution or hypertonic situation, and that's going to cause shrinkage of the red blood cells. So in a region where there is high solute concentration of the plasma, the red blood cell will go from this to this. It's going to shrink. Now if the roles are reversed and then the plasma water content is extremely high or too high, higher than the water content of the red blood cell, the reverse is going to be the case. But water will still move from a region of higher concentration to a region of lower concentration, meaning that water will still move from the plasma into the red blood cell, which will cause expansion of the red blood cell, and this is going to become, in a hypotonic situation or hypotonic solution, this is going to be what our red blood cell looks like. And this is very, very important in cases like diabetes mellitus. Okay, this is very important to note, but just hold on to this information for now. It is very, very vital. Okay, so that's all for our passive transport or passive diffusion. Now we're going to go into active diffusion. So active diffusion, now let's go back to these three key things that I spoke on earlier. Number one in active diffusion is that there is going to be use of energy. So this is for passive, and then this is for active transport. Okay, so number one, there's going to be use of energy. Number two, instead of moving down or along concentration gradient, in active transport it is moving against the concentration gradient. So that means if it's moving like this, it is moving like this, or uphill. If the gradient is going like this, it is going to go like this, and then it is going to involve, so active transport always involves the carrier protein. Okay, so now the divisions under active transport. So active transport can either be primary active transport or secondary active transport. So in primary active transport, okay, now this is very, very important. Please pay attention, because this is very important. What we're going to be talking about now, you're going to build up on it throughout your med school journey, and it's also very important in your professional exam. So under primary transport, one of the examples of primary transport is sodium potassium ATPase pump, or sodium potassium ATPase pump. So let's come over here, please. Okay, so in primary type of active transport, number one, the energy is gotten from the diffusing molecules, or the energy is gotten from the molecules that are being transported. So let's take sodium and potassium for example. Now let's just use this. Just ignore the calcium, but let's just use this. So when primary active transport wants to take place, there are three major and then three very, very important steps. We are going to call them BDC. BDC, this is the mnemonic I created for it. It's also the step, was the mnemonic that I created for it. So first of all, now in the transport of sodium and in transport of potassium, That's in action potential. Sodium is going in and potassium is going out. Sodium is going in and potassium is going out. So for every 3 molecules of sodium that is going in, 2 molecules of potassium are going to be pushed out. Okay. So this is inside the cell and then this is outside the cell. So now BDC what explains what happens, is the mnemonic that explains what happens here. So number 1, B is for binding. So now the sodium is going to bind to this part. So this is a channel. Okay, this is a channel, which means it is an integral protein. So the sodium is going to bind to this part of the channel, okay, and it is now going to cause a reaction, and that reaction is called a dephosphorylation reaction. So the D is for dephosphorylation reaction. And what happens in this dephosphorylation reaction? ATP is converted to ADP and pi. ATP is converted to ADP and pi. That pi is inorganic phosphate. Okay, so once this happens, the conversion of ATP to ADP and pi, there is release of energy, there is energy generated, and then this energy, so it's going to, so this reaction, okay, it will take place here, and then it's going to cause a conformational change. Conformational change is going to cause a conformational change here, and it will cause this channel, this our gated, because it's gated, it's going to cause this channel to open and it will become like this. And it will let this sodium go in and then close again. It's a hinge like motion, like the motion of a door, a hinge like motion. And after this sodium will go in and then it will now return back. So it will enter into the cell, it will be able to enter into the cell, and then this motion, this door, will be able to return back to its normal state. Just explained how primary active transport works using the sodium potassium ATPase pump as an example. So 3 sodium molecules in, 2 potassium molecules out. Okay, now the second one we're going to explain, that's the second division under the active transport, is the secondary active transport. So since we already understand under the primary active transport, it's going to be easier to cope with the secondary active transport. So under secondary active transport, we have the syn port and the counter port. Let me just bring here out. Syn port and the counter. Let's call this counter transport. This is very easy to keep in mind. You can just say syn port, same port, syn port, same port. That is okay. In the secondary active transport, the transport is based on sodium. As explained before in the primary active transport, sodium moves in and potassium goes out. So in secondary active transport, the molecules that go in with sodium are called syn port. Like the transport, if the molecule is going in with sodium, it is called syn port. But if sodium is going in and that molecule is going out, it is called counter transport, like counterclockwise, against or the opposite side of the hands of the clock. Counter transport, opposite of syn port. So for molecules that use the syn port transport or are transported via the syn port secondary transport, they are called, an example is glucose. Glucose is moving from a region of lower concentration to a region of higher concentration, so it's called glucose. And then there's the counter transport example is via calcium. So this is almost the exact same thing we just explained here with the sodium potassium ATPase pump, but we're using calcium. Calcium is, this arrow means higher, so calcium is higher here outside the cell. Now as sodium is going in, calcium is trying to move out. So this is the region of higher concentration. Remember, I said it's moving from a region of lower concentration to a region of higher concentration. So the calcium, because of the reaction, the binding, the reaction, calcium move out, Pi is released, energy is generated. So in secondary active transport, the energy generated is from the transport of sodium and potassium pump. Like the molecules being transported, the molecule or molecules being transported, they derive their energy of transportation from other molecules that are being transported, like sodium in the sodium potassium. Okay, so now we're going to talk about vesicular transport. Remember, divided into A and B, molecular transport and membranal transport. And membranal transport is vesicular transport. So vesicular transport is for transporting large proteins. Large proteins being that, number one, they cannot pass through the pores. So in the cell membrane, the bilipid layer, there are actually some little pores which allow some molecules to pass through. But since these proteins are large, they cannot pass through. Wait. So under vesicular transport, the first type of transport we have is endocytosis. Endocytosis is going in. Endocytosis, the mechanism is infolding. So whatever is going in, like ingestion, that's endocytosis. So examples of endocytosis can be pinocytosis. So pinocytosis is called cell drinking. It's in little. The vesicular transport here is for in comparison to the other types, is for little particles. It's pinocytosis, cell drinking. Then we have phagocytosis, which is cell eating. So phagocytosis is different from cell drinking, being that there are some cells that actually have this function. Okay, their entire function is this particular phagocytic action. An example of this is called macrophages. The type of cells that have this kind of function are seen in the immune system to remove debris, to engulf debris, bacteria, or any foreign microorganisms that are not supposed to be found wherever they are found. So the process of the phagocytic action is divided into AT, E, and DE. AT, E, and DE. So 8. Phagocytic action is cell eating. 8. So, and you can just put the DE. So now, I started explaining this before. If this is a cell, this is just a normal cell. Let's say this is a macrophage. Let's say this is a macrophage. Now, this is going to stand as bacteria. Let it stand as bacteria or debris, but right now I'm going to take it as bacteria. So now the first step in the phagocytic action is attachment. That's what AT stands for, attachment. So this macrophage, okay, is going to attach to the cell body or the outer layer of whatever microorganism or debris this is. Okay, it's going to attach to it. Let's bring it out. This is the cell, so it's going to attach to it. So then the next thing that happens, E, is engulfment. Sorry for my handwriting, but engulfment. So what happens in engulfment is that as the cell membrane has attached part of it, as the cell membrane has attached partly to this organism, okay, there is a motion, okay, so part of the cell itself, like the inside of the cytoplasm, is thrown out to cover this other part of the bacteria. So as this part is attached, this part inside the cell membrane is thrown to cover it. So that's actually the engulfing motion. It's like I'm holding this. Okay, let's say this is it now. So I'm using this side to hold it, and since my mouth is part of my, let's say I'm an entire cell, okay, my hands are the cell membrane, I hold this part like this, and then I will throw myself out and engulf the entire bacteria. Okay, engulf the entire bacteria. While this is happening, okay, this part breaks down, like the fusion part breaks down. That means that what's going to happen now, will now be that this will be the cell, this will be the nucleus, and then the debris that just got engulfed, because this part is going to be thrown out, okay, thrown out in a bid to surround it, okay. The entire function or the entire aim is to surround it, and then this is going to be, the bacteria is going to be engulfed into the cell, and then DE is for destruction. So the destruction can either be, as discussed earlier, due to hydrolytic enzymes, of the lysosome, or due to the oxidative the catalytic enzymes or catalase enzymes of the peroxisome. So what does that mean? That means that macro, so whatever we learn, we are supposed to relate it to what we already know. That means that macrophages, or cells that practice phagocytic action for the purpose of destruction, for the purpose of protection of the body, they have good or higher concentration in lysosomes and peroxisomes than other cells around the body. Okay, so that's phagocytic reaction. Then receptor mediated transport. So as discussed earlier, or from what I've said earlier, majorly on the cell membrane, okay, on the cell membrane, the molecules that serve mostly as receptors are actually the proteins. In some instances, yes, the glucose part can also serve as receptors, but most of the time is the proteins. So in receptor mediated transport, now there is going to be a molecule, let's say iron, Fe for iron. There's going to be a molecule outside, okay, and then it will bind to the receptor. Obviously there will be specific receptors for this particular structures. There are some structures that can only enter the cell through receptor mediated transport. Okay, so there's going to be a type of receptor for iron, okay, and then that will now, it will recognize iron. Receptors, they are concentrated, they are specific for a particular substance. So these iron receptors on the cell membrane are specific for only iron. So once they bind to iron, they will open and they will be transported. They will open and they will be transported. So apart from iron, another substance that can be transported via this mechanism is cholesterol. So that's all for receptor mediated under endocytosis. So now we just finished discussing endocytosis. The method for pinocytosis and phagocytosis under endocytosis is infolding. Infolding, whatever it is that we are trying to bring into the cell, is for things that are coming into the cell. Infolding, so like a hug, infolding, entering the cell. Now the second one we're about to discuss is exocytosis. Exocytosis is taking out or pushing out. Exo, endo. So under exocytosis, our mnemonic is MFB. Let us bring it out here. MFB. So now this exocytosis, since it's a method for whatever is inside the cell to come out, that means it can be used to bring out, or it can be used to expel waste, or it can be used to, the important function which exocytosis is actually used for is for cells that actually secrete, for secretion of hormones, So now the mechanism of exocytosis, let's get down to the mechanism. So the M stands for migration. Now what does this mean? This means that as inside the cell, inside the nucleus, okay, the ribosomes come outside, they come into the rough endoplasmic reticulum. In the rough endoplasmic reticulum, protein is synthesized. The synthesized protein goes into Golgi apparatus to be transported. Now that Golgi apparatus migrates, that is that word migration there, it migrates to the cell membrane. Inside the cell, if this is a big cell, that Golgi apparatus carrying the protein, a vesicle, it is now a vesicle, it migrates to the cell membrane. It just gets here. So let's say this is an entire cell, it migrates to the cell membrane. Then F is for fusion. So when the Golgi apparatus gets to the edge of the cell membrane, it is carrying proteins, whatever, maybe, proteins, hormones. Most hormones are proteins, but not all are proteins. Most hormones are proteins. So the the Golgi apparatus, as it has migrated to the edge, it now fuses to the cell membrane. Fuses to the cell membrane like this. So once it has migrated here, it now fuses to the cell membrane. Okay, this is the membrane. This darker one here is the membrane of the vesicle, and then this lighter one here is the membrane of the cell itself. Then B stands for breakdown. Breakdown. So what does that mean? That means that the area of fusion between the cell membrane of the Golgi apparatus and the cell membrane of the cell, let's say they fuse, and then the area of this particular place here, the area of fusion, that area of fusion between them will break down. That area of fusion between them will break down, and it will look like this. And when this happens, what does that mean? That means that the particles that were transported, the particles in here, will be released out into the extracellular fluid. That is the fluid surrounding the cells. Extracellular fluid outside the cells. Intracellular fluid inside the cells. The same way the extracellular fluid of the red blood cells is plasma. This is all, everything is related. What you are learning now is going to be built on throughout your entire stay in med school. So these proteins, hormones, are going out. They will be expelled out via this mechanism, and then after that the membrane is going to come back together. Great. So now via this we've been able to expel out. Exocytosis has taken place via MFB, migration, fusion, and breakdown. Now this exocytosis, the process of exocytosis actually depends on some particular things. Number one, DP, docking proteins. Docking proteins. Now in another video we are going to be going especially into a type of cell that majorly mostly functions on exocytosis.
Today we were able to cover transport across the cell membrane at the molecular level and at the level of the cell membrane. I hope you were able to understand this video perfectly. If you weren't, you can always write in the comment section whatever your questions are, or save this video and download it for later. We are Dicot is fam. Please don't forget to like, subscribe, and forward this video to whoever needs it. Bye.